Good morning, our students. Welcome you all, and we wish you happy and glad and succeed in this stage. Now, our lecture today is pertaining out concerning the helminths worms. Now, the classification of these worms can be shown in this slide. It is belonging to subkingdom Metazoa, divided into phyla Nemathelminths and Plathelminths. The Nemathelminths first phylum can be divided into two classes: class Nematoda, roundworms, common name roundworms, and class Filaria, tissue roundworms. And the second phylum is Plathelminths, also having two classes: Cystoda, tapeworms, and class of Trematoda, Flux, which is the lecture of that today. Other slide. Now about general information that concern trematodal parasite. You should know and you should imagine in your mind the information that concern each parasite. As a whole, in general, the trematodal parasite having many criteria represented it. So the first one is they are commonly referred to as flux. Other name is flux, trematoda or flux. The life cycle having three morphological forms. The first is egg. Second is multiple larva stages, maybe one larva stage or second or other. And the third stage is adult worm or form. The egg, which are the eggs, which are the primary morphological form recovered in human specimens. They are very. Some contain operculum, cover-like, and other distinguished by the presence of spines like schistosoma groups or trematoda or flux of blood flux. Now, other criteria is the larva stage typically occur outside the human host. Other criteria, the adult flux are thin or non-segmented, resembling leaves both in shape and thickness. Each adult flux is equipped with two mussel or cup-shaped suckers, one oral, called oral sucker, and the second located ventrally, called ventral sucker or acetabellum. The life cycle placed into two categories. Those as a whole, we mean as a whole for all, those take up residence in intestine, bile duct, or lung, which mean organo-dwelling trematode. The first. Second category is those take up residence in blood vessels around the intestine or bladder, which mean blood-dwelling trematode. This concerns the schistosoma species. Now, a human infection of organ-dwelling flux occurs following the ingestion of water plants, fish, crab, or crayfish contaminated with encysted metacercarium. This for the group of organ-dwelling. The infective state is metacercaria, and by contamination of the latest larval stage or its intermediate host, which is plant or fish or crab or crayfish. Now, human infection with the blood, whereas the human infection with blood-dwelling flux occur following the penetration of cercaria into skin by infection of cercaria, not metacercaria, and penetration of the skin. Now the phylum of Platyhelminthes, special class trematode include intestinal species, and we can give you an example like Fasciolopsis buscae and Metagamous ekagawa and Heterophis. Second, liver species, example Fasciola hepatica, that's the lecture of that today, and Clonorchus other species. Third group is, or class, lung species, example as Paragonymous wistermani, and blood group or species include Schistosoma species, which are Schistosoma mansoni, Schistosoma japonicum, and Schistosoma hematopium. This slide can show you the general life cycle of the flux or trematode. Well, the eggs are released from human in feces or urine in fresh water in environment, maybe in feces or in urine as the species. Now, egg hatch in first larval stage, which is larval stage, miracidial larva that invade a snail for as a first intermediate host. Here the snail is the first intermediate host, and the hatchet stage from the egg is miracidial, free living miracidium. All trematode except Schistosoma species require a second intermediate host, which is mostly a water plant or animals. The organ-dwelling trematode need more than one intermediate host, but at blood group Schistosoma species having one intermediate host. It is different. It is different from that. Now, the second larva stage, cercarial or metascale larva, come out of the intermediate host. Now, about the infection of the human can occur through direct penetration of cercaria as Schistosoma species, or injection of encysted metascaria in water plant or animal that concern or other trematodes. This slide can show you the medically important flux. You know, the trematode belong to the Platyhelminthes, and they have a wide range of habitats. The great majority inhabit the alimentary canal, as we show you before preceded, and liver and bile duct, ureter, and the bladder of vertebrate animal. So that, and accordingly to the site that they inhabit, there are 4 groups of flux. These are as follows: first, liver flux; second, lung flux; third, intestinal flux; and fourth and last is blood flux. Now, the first species here that represent the liver flux is Fasciola hepatica. Fasciola hepatica, its common name is sheep liver flux. Now, this first species represents the liver flux, and it is with severe infection and can disease the sheep and the human, and can cause cost, healthy and economically. So Fasciola hepatica, nematode, are not thought to infect man, but in fact man is not an unusual host, with infection being reported in many countries including Europe and the USA. The eating of unwashed watercress appear to be the source of infection. The source of infection is unwashed watercress because it harbors the infective state, which is metacarrier, and with then ending up in the liver. The most common host is sheep, where they can cause severe disease. Now, about its life cycle and the transmission routes. How can it complete its life cycle and how can it be transmitted? The life cycle of this parasite and other species, as we noticed before, Pleuronochus sinensis, which related to the liver flux, are complex. We mean it requires more than one intermediate host. The adult worms inhabit the liver or bile ducts of the definitive host, a human. Is a human. Now, where they lay many eggs, which are deposited in the environment in feces, not in urine, in feces. They are immature when passed out, when deposited out, discharged out in environment from human, infected human. They are immature. If they are passed into water environment and touch the water, they become mature in 9 to 15 days at optimum temperature of 22 to 25 degrees. This slide can show you schematic representation of the life cycle of the sheep liver flux. We will start from the adult when be found in human liver or biliary duct. The adult can give or discharge an embryonated egg in feces, in biliary ducts, then to GI, then to with feces to be extracted out. Now, an embryonated egg in feces. An environment here, an environment that having snail and water plant as intermediate hosts. An embryonated egg is embryonated egg. The embryo, it will form and develop it in the egg. Now, from embryonated egg, the miracidium stage, first stage hatched from the egg is miracidium. The miracidium free living avixilia, pili, and can infect and enter snail. Snail is the first intermediate host. Now, the miracidium stage can develop it to other stages, which are sporocyst, androidea, and cercaria. Now, the cercaria with its tail be discharged from the snail, first intermediate host, to be free. Now, and be insisted on water plants as a second intermediate host. Stay at the water plants as a metacarrier. Water plants can harbor the insisted metacarrier. Now, the human come to ingest vegetation, aquatic vegetation, which is contaminated with the metacircarium, so the infection will occur. Now after swallowing it, the exists in the duodenum. In the duodenum, the metacircaria, encysted metacircaria will be exist and attached to the mucosa of the small intestine, and then penetrate, and ultimately it will the liver and biliary ducts. Okay, in general, the life cycle of fasciola hepatica can be stated as follows. The immature eggs are discharged in biliary ducts and in the stool, from biliary ducts to GI, then in the stool. Egg become embryonated in water. Egg release melacidia, which invade a suitable snail as a first intermediate host. Now, including many species of the genus Lima, as a species, as a name of snails, we can't concentrate on it, but just you should keep in your mind the first intermediate host is a snail. Okay. In the first intermediate host, the snail, the parasite undergoes several different stages, which are sporocysts, vidia, and cercaria. Cercaria are released from the snail, the first intermediate host, and insist as metacercaria on aquatic vegetation or other surfaces. Mammals acquire the infection by eating vegetation containing metacercaria. A human can be infected by ingestion metacercaria containing freshwater plants, especially watercress, and after infection, the metacercaria exist in the duodenum and migrate through the intestinal wall, the perineal cavity, and then to liver parenchymal and to the biliary duct, where they develop into adult worms or foam. In human, maturation from metacercaria into adult flux takes approximately 3 to 4 months, and the adult flux fasciola hepatica is up to 13 micrometer by 13 micrometer and reside in the large biliary ducts of the mammalian host. Now, the fasciola hepatica infect various animal species, mostly herbivores, as sheep. Now, about the morphological criteria or feature of the adult worm is represented here in this slide. The morphology of the adult flux of fasciola hepatica is well documented. They are large leaf-shaped parasite, like the leaf, resemble the leaves of the plant. Leaf-shaped parasite about 2 to 3 cm long. They are having 2 suckers, an intraoral sucker and surrounding the mouth, and a ventral sucker acetabellum on the ventral surface. And the outer tegument or cover or membrane of this parasite is covered in tiny spines which face backward, enabling them to attach themselves along with their sucker to the tissue, help to the attachment. The suckers, oral sucker, ventral sucker, with the spine that cover its tegument, can help this parasite to attach themselves to the infected tissue. So its function for attachment purposes. Now, about the second stage of this parasite, the ova or ovum singular, Ova are thin shelled, ellipsoid, quinine colored with yellow color, barrel stained, with an operculum. Here the operculum cover like that is often inconspicuous, difficult to see, but it is found. The egg is ellipsoid or oval shaped and barrel stained, having operculum inconspicuous with operculum. The ova of fasciola hepatica are ovoid in shape, one in color, and often showing an inconspicuous operculum. It's very important note, which is there is much crossover in oversize between all of the fasciola species, so we can not depend on the phenotypic criteria of the stages, this stage, to differentiate between the tematodal species that can cause disease, but we should pay attention and concern on the dietary programs of the patient and case history and region that be epidemic or endemic from this parasite. All this information help us to give an idea and good mapping on the persuaded cases here to send the patient for do the best lab diagnosis to present the case. Now about its pathology and clinical symptoms. Here in this slide, the transit of immature worms through the liver can cause mechanical and toxic irritation with toxemia, necrosis, and secondary fibrosis. The development in the bile duct can cause cystic enlargement and endothelial hyperplasia and adenoma. Also, secondary inflammatory infiltration causing fibrosis and cholangitis. There can be secondary bacterial infection causing abscess. The other market is Eugenophilia. Here, any parasitic infection, especially by worms or helminths, can rise and elevate the level of eosinophil cell. So the Eugenophilia is a market. Worm can appear ectopically in lung, brain, eyes, and other sites with similar reaction. If raw sheep or goat liver infected by the adult flux is eaten, there can be local irritation and pharyngeal infection. So this adult worm can be present not in human being only, but in other herbivorous animals. And if they infected their liver infected and eaten by a human being, it can cause local irritation and pharyngeal infection. About the infection, in acute infection, it may present with fever, represented or the patient symptomized fever, tenderness, hepatomegaly, epigastric pain, anorexia, and vomiting. Jaundice may be occur. This in acute in acute infection. But in chronic infection, when prolonged in time, there are maybe not asymptomatic. And you know why asymptomatic? Because there the dose of parasite is less or few and the high level of immunity of the infected ones and the various factors of the parasite. All of this can bring about why the individual cannot be symptomized, why the infected ones cannot be symptomized. Now, or epigastric pain in our up side or chin, light upper gluten pain, hepatomegaly, and vomiting. All these symptoms related with chronic infection. Okay. Now the last slide here is laboratory diagnosis. Any persuaded case come to you as a physician, you should keep in your mind, firstly, you should know the case history of the patient, clinical symptom and site, and dietary program, and epidemic level, journal. All this information should be kept in your mind before send your patient, admitted your patient or your patient to the lab. So, you know, this adult parasite can survive or dwell the liver and biliary duct. So, we should keep in our mind and our thought, we should use the stool sample to be examined in lab. We send the patient for lab for stool examination. So, definitive diagnosis is made by observing the ova in feces here, because this stage can be excreted out from GI with stool. Since the flux are very prophylactic, any significant infection will be easily picked up, where identification cannot be made from the size of the ova. Clinical information and the source of infection may help to provide a diagnosis, as we mentioned before. Now, if this parasite can be present ectopically in other sites, organs, liver, heart, eyes, so the serological techniques are available for diagnosis of fasciola hepatica. My study